All right, hello everybody. I will get started in a few minutes, but welcome to your online class. I'll wait for the rest of the group. Say hello if you're here. I can't see who's watching me right now. So the second part of the class is actually going to be on Zoom. I put the link for Zoom into the put the link for Zoom into the announcement. So let me just pull it up right now so everyone can see where to find it. Go. Hi Jessica, hi Beatrice, hi Jasmine. Okay. So here in the announcements is where you would click to join the Zoom meeting. That will start at 3:30. Okay, so this is where you watch me give my lecture. Okay. So again, a couple of reminders. Your Blackboard quiz will be up soon, and that will be due on Saturday. Okay, so we're getting a slow start to the week, but hopefully this won't be too difficult. I did see that some people already did send in their research topics. If I thought you needed to change it a little bit, I gave you some comments, but as long as it was a reasonable topic, I gave you full credit. If I thought I only docked a few people, but if you turn it in again, I can give you full credit. Okay. And a reminder that if you go to course content, no, not course content, sorry. If you go to, well, that just exited me out altogether. All right. A reminder that if you go to syllabus and schedule, you will get to this introduction to the online course and a recording of me going through a few pieces, okay? So, okay, good. I'm glad you found the sound, Jessica. Okay, I record all of these and I will be putting it up onto Blackboard. So after this, if you have any connectivity issues, you just go to the lectures, your lecture PowerPoints, and what I'll do is the online lecture here. I have these two linked here, okay? So we have the scheduled Twitch stream there. The video is going to be posted there. Chapter 9 and 10 may be included in the Twitch TV stream here, but I will probably not finish this until Thursday. And so that's going to be posted on the under there, okay? And as we move forward, there's going to be more. Okay, So let's move and start this. Now, chapter 9, I mentioned before, we are actually going to be, we'll start from chapter 8. Sorry, I didn't pull it up like I wanted it to. So we'll be doing chapter 8 as well today, but we're finishing it up pretty quickly. I don't want to... not what I want to do. What are you doing? It's very confusing what is happening right now. Okay. Exit show. Now, this is what's happening. I accidentally... Okay. Apologies, this is my own issue. Okay, this should open properly now. There we go. Okay, my fault. I have to set up the show too, so you can see it on my screen properly. Okay, so you've already gone through some of chapter 8 before. We will be doing some of chapter 8 right now. Yes, all the PowerPoints are actually already up. If you go to the online lectures in lecture PowerPoints, the PowerPoints are up. And I'm just going to be posting the lectures of me doing them, okay? I'm anticipating I'm going to go reasonably slow. The major goal is that we get through chapter 17, so I may chop up some pieces. I really want chapter 17 to be part of this class, and that's right now at the end of class, so I may chop up a few of these into smaller bits later if we start running out of time, okay? But for sure, I am going to try to get through chapter 12 before our second exam. Okay? All right. So we already went through a few of the pieces about how bacteria work. They have a singular circular DNA molecule. It is their chromosome, but it's not structured like our chromosome. And they have these extra chromosomal plasmids that they can use to turn on sometimes. And that's where some of the interesting pieces about bacteria come through. So this has to do with recombinant DNA studies. And again, we're going to be going through a lot of, in the case studies in particular, introducing that pretty fast, the recombinant DNA. And we'll be working through the pieces in the lecture a little slower than the actual case studies. All right. We're going to move past some of the serial dilution. So genetic recombination in bacteria, we are able to recombine different pieces and chromosome map. So we can replace genes present in the chromosome of one cell along with the chromosome of a genetically distinct cell. And this is where GMOs come from, basically. So this is an interesting piece. Sorry, I'm making this bigger. And that's really why I wanted to introduce it now, because we're going to be going through a lot of genetic recombination things before we get back to the actual, here are the techniques of genetic recombination. So in eukaryotes, it's crossing over, but in bacteria, this is recombination. But genetic information is transferred. It is a little bit different than eukaryotes and sharing genes. Because bacteria don't fully sexually reproduce because they don't do meiosis, they don't have gametes, but they do share and mix around their DNA. Okay? So we went through this before, but genetic information can be transferred from one bacterium to another. This can result in an altered genotype, basically sharing some of those extra plasmids. There's conjugation, transformation, and transduction. And so this leads to different types of gene transfer. So vertical gene transfer happens between members of the same species. Horizontal gene transfer is transfer of genetic information between unrelated cells. Horizontal gene transfer, because of the way the bacteria is, they have very loosey-goosey kind of sharing with whoever they want. That means that horizontal gene transfer can change the way that species outside of bacteria can be altered, okay? Antibiotic resistance genes to another species, and it's a major factor in speciation. This is why if one species of E. coli, if one species of E. coli gets resistance, that's why everything can get resistance. Basically, in between multiple bacteria, they can share resistant genes because those are the little plasmids they have in there, okay? So in horizontal gene transfer, they can do this through conjugation. Direct transfer of DNA, basically they have to be touching to do this. Sort of the bacterial version of sex. And transformation, bacteria can just like take up free DNA. And you do this in Bio 1140. Again, we had talked about this before. Bio 1140, you transform some bacteria by putting these free plasmids into a solution and encouraging the bacteria under stress to take up the plasmids and incorporate it into their own DNA. Bacterial viruses can also take DNA from one bacteria to another. We're going to be talking about viruses a whole bunch. But, because, well, it's very relevant today, huh? So, overall, we have a donor cell and a recipient cell. They make a cytoplasmic bridge. Again, that's the touching, that's the conjugation. And the DNA replicates and then transfers a chunk of its DNA, not all of it, to another cell. That transferred DNA then crosses over with the bacterial chromosome, and you create a recombinant chromosome, okay? The transformation, which again is what you did in class in Bio 1140, there's free DNA in an area. Naked DNA is taken up by the recipient cell, usually under stress, and then the crossover happens and it replaces part of the bacterial chromosome with that bit of the chromosome. Transduction is when this is a bacteriophage. Bacteriophages attach to the bacterial cell. They inject their own DNA, and that DNA uses the bacterial information to replicate itself. And then it makes the bacterial protein through RNA and ribosomes in the DNA itself. Those new viruses then infects a new bacterium, and they carry the bacterial DNA, and they move, and as they infect it, they can lead to a new recombinant chromosome. So this is sort of a weird way of creating a new recombinant chromosome. The bacteriophages can carry bits of the DNA from one bacterial cell to another. All right, so we talked a little bit about conjugation before, but it's not necessarily reciprocal exchange. This is just one transferring to another, okay? All right, unidirectional transfer. We have the donors and the recipients, and these fertility factors. Okay, so that we discussed this a little bit already. What I wanted to focus on is this, okay? So this is sort of one way that recombinant DNA in bacteria can be made. So I'm just going to walk through kind of the process here. I'm not going to go to this is from your book. I think it's a little confusing. This is from a different book, and I think it is a little bit clearer about this. Often, these may not even share their actual chromosomal DNA. Instead, when they connect to each other, the plasmid itself duplicates, and then one strand of the plasmid turns into a second one. And so this is horizontal. It could be horizontal, or it could be, it could also be vertical. But basically, any bacteria that are together that have functioning the pylases that can go together, they can replicate. Okay. So this one now has the plasmid gene. So these are, again, these plasmids represent things that it often uses in periods of stress. It's not the vital genes. they can transfer that information from one to another, and this can be something like a antibiotic resistant gene. And this can make ones that aren't antibiotic resistant in the presence of even dead things. They can pick these up through that picking it up inside of their cells, or they can actually transfer that information between cells. All right, so this is I've been playing Pokemon all week with my daughter. This is a Pokemon thing where Staphylococcus aureus is evolving into methicillin-resistant Staphylococcus. It's an interesting pattern of the way that resistance is passed on. It's not passed on necessarily directly from one bacterial generation to the next. It can be passed on by just being near other Staphylococcus bacteria that have the resistance gene. All right. So cell contact is necessary for chromosome transfer to occur. If there's no direct contact, then these things called auxotrophs do not exchange genetic material, and you don't have the prototrophs, the ones that have the new cells. They are not formed. Okay, so physical contact is necessary. So these high frequency of combination cells, what it does here is that this is a way HFR cells are often used in bacterial recombination in the lab, where basically these bacterial chromosomes are more likely to cross over with the F factor, these external plasmids, and integrated into their cell, which means that you're more likely to get extension of the bacterial chromosome. Okay. So these cells behave as F plus cells, and they undergo conjugation with F minus cells, but they can also they cross over and can incorporate it into their own DNA into their major chromosome instead. Okay. So I wanted to point this out because this is an interesting piece. How long they conjugate these high frequency cells and the F minus cells, how long they conjugate depends on how much of their genome is transferred. If you cut it off in between, think about having a backup on your machine, and you cut that backup off, you've only transferred some of the data. And so this is a way that you can actually map bacterial genomes by taking this HFR cell and trying to figure out where along the bacterial chromosome the different pieces were transferred. Okay, so if you watch this, okay, so conjugation is started. This F factor is nicked, and the 5N moves into the F minus cell. Okay, that's the conjugation transfer. And then the transfer strand is replicating while this is happening. And then later, this HFR chromosome, I'm supposed to turn that off, sorry. I'm just going to quit this. So later, this other gene gets moved in. And then later, the crossing over can lead to the recombination of alleles, and this one can take up certain things from the chromosome that has been transferred. So all of this to say that you can map bacterial genes with interrupted conjugation. Okay, I don't need you to remember all of this. 2F plus cells, F minus cell becomes F plus. This is beside the point. More what I want you to understand is kind of that you can map bacterial genes with this interrupted conjugation. Figure out what's first, what's second, what's third. And so this is an interesting, different way of mapping chromosomes. Bacterial chromosomes are the only ones that this works But okay. So it takes about 100 minutes to fully transfer the entire E. coli chromosome from a high frequency recombinant cell into an F- cell. If conjugation is interrupted, then only part of the chromosome has been transferred. So distance between genes can be determined by measuring the time required for different genes to transfer from the HFR cells to the F- cells. Okay, so this tells you how far apart are the genes. This is a way of doing it without with all that crossing over. Again, everything is on the same chromosome in bacteria, but now we can map bacterial chromosomes. Okay. See. Oh, well, that's not working. I'll see if I can get you a better animation here. I'll post it later with chapter 8. Okay. But here's the concept. Okay. Interrupted conjugation can be used to map bacterial genes via this. Okay, so we have an HFR cell with a certain genotype. We know the genotype. We can sequence the genotype, but it is pretty difficult when sequencing it to actually figure out, we can find this gene and this gene and this gene, but the actual distance and the and how far it is. Okay, so we can see all of these traits in it, but knowing how far apart they are is difficult. So what you do is you start a transfer with this HFR that has it all incorporated into its circular one here. You start the transfer, okay? And then you stop it. So you have this 0 time where it first started. You see what was first put in. Okay, so these are very close together. LEU and THR, okay? And then 8 minutes. You have these, you separate the bacteria, and now you have these two and this new one, okay? So this must be a certain distance away from these two, okay? And then you separate them again at 10 minutes. Well, now they have this new gene. And then you separate them at 16 minutes, and then you separate them at 25 minutes. And now you can get this clear, linear progression of genes and know which one is first, which one's second, and which one's third, okay? So this is a way that you can map bacterial genes. You can't do this with human genes because, or the things with chromosomes, bacteria don't have true chromosomes. They just have a sort of an uncoiled, loose DNA. And so you can't actually do this with all the chromosomes, but this is one way that you can map bacterial genes using all this information about the transfer rates, because it transfers at a specific rate. And so now you can measure how far apart they are and what their order is. Instead of just observing that they have all of them, find out where they are on the bacterial genome. This helps us to tag certain genes and helps us to track when to expect them to transfer and can control different bacterial recombinants. Okay, I know I went through that part a little bit fast, but this is the major thing I wanted you to get from this. Does anyone have questions about understanding why the recombinant part, bacterial conjugation, can be used to map genes? I'll wait for a second. I know I'm at a 4-second delay or something. Nobody's here? Okay. Okay. Well, I did upload it. So Faye, I did upload it onto onto Blackboard. It's definitely there. So if you're having trouble accessing it, I could check and make sure, but not sure. I will double check that this specific one is uploaded. Perhaps I uploaded an old version. I'll double check that is correct. But this will definitely all be up right after. Oh, not right after, but soon after, okay? All right. But this is one of the things that you're going to have to solve, okay? So if you have this information, you can then tell me, how would you do this? How can you map the genes? What kind of patterns would you expect, etc. Okay? So the transfer times indicate the order and the relative distance. Okay, so what you can see is that these two are very close together, that they both transferred. The percentage of cells displaying a particular trait, most of them transfer really quick here. Okay, and then these lagged behind, and then this one lagged behind, and because each of these were transferred later, 10, 20, and 30 minutes to get each one of them, it tells you how far apart they are. So this is just a genetic map. Again, it's still relative distance, but it gives you an idea of this one is a few seconds apart, therefore it's a few units apart. Okay. Well, that's good that, Beatrice, you see that it is working for you, so that's good. Again, you can watch this again. Please do watch it again. If you wanted to study, you can fast forward me. If you're bored by me, you can focus in and make me repeat things over and over. This will be posted on YouTube shortly after we finish our recitation. All right. So, we're going to shift a little bit. I just poured a pile of information about bacterial mapping into here. I wanted to go back to this concept of horizontal versus vertical transfer of genes. Okay, so horizontal transfer, again, is the transfer between two unrelated cells. Well, how does that work in terms of horizontal gene transfer in larger organisms? So let's look at some interesting stuff, okay? All right, scientists discovered this sea slug. So, what is interesting about the sea slug? What does it look like? Anyone want to type it into the comments there? What does the sea slug look like? Okay, yes, it looks like a leaf. Excellent. So it looks like a leaf, and this is actually reasonably normal for this kind of slug. Now, the question of why, scientists thought they knew why. Because many other slugs related to this green sea slug are shaped like plants. Elysia crispata is a lettuce sea slug. Elysia diomedia, well, it looks like a piece of kelp here, okay? To get color throughout their body, they have a heavily branched digestive system. Basically, their digestive system goes through their entire body, and as they eat the things that they hide in, it colors their body. Food is deposited there, and that causes the color. So, at first they were like, oh, this is a very green version of this Elysia slug. We've seen these before. They use algae to incorporate into there. But something was weird with this slug. This sea slug was green, but really green. Like, more vibrant than any of the other sea slugs. It's almost too green. They were thinking, it looks like the algae is alive inside of the bacteria, inside of the slug. Well, that would be weird. Normally the slug is eating the bacteria, eating the algae to get this into their bodies. Okay. Researchers thought perhaps this is a weird symbiosis. We haven't seen this before, but the algae is alive in the slug, and the slug is getting some of the algae's stuff from the slug. Okay? But then they looked at it under the microscope, expecting to see living algal cells inside of the sea slug. Instead, they didn't see algal cells. They saw chloroplasts. That's super weird. Chloroplasts shouldn't exist in an animal. How could chloroplasts survive without the surrounding algae? That's a good question. Also, why are the chloroplasts there and not the rest of the cell? What is the sea slug doing with those? Because the chloroplasts weren't just there, they were alive. They were working. They weren't broken up into pieces and just the green color remained, which is true for many of its relatives. Instead, they were actively alive and working. All right. This is super weird because even if we were able to take chloroplasts and put them into our cells, make our skin green, we don't have the enzymes to keep them alive. They need specialized enzymes. So something strange is happening here. This sea slug was dubbed a kleptoplast, literally a plastid stealer, stealing the chloroplast from its algal food. So what's the next question we should be asking? Now that it has the chloroplasts, what should we ask? Is it actually photosynthesizing? Is it using the chloroplast, not just keeping them alive, but actually actively turning sunlight into sugar? So that would be crazy because it's not a plant. Okay, I flashed on the screen, but so how would, I guess the question I wanted to ask you guys is, how would you set up a experiment to figure out whether or not this sea slug is indeed photosynthesizing? Any ideas for how to set up a experiment to figure out, is it photosynthesizing? Is it actually using the chloroplast, and not just for color, but actually for food? That the chloroplasts are giving it sugar. Any ideas? How do we set that up? Well, my stream's still working. Just, you guys are thinking? Definitely have at least 6 or 7 people in here. Okay. So, Ifani wants to put one in sunlight without excess food. Yeah, that's good. Now, what do you expect if you do that? Instead, here, also you could isolate the sea slug. I'm not sure who the limit breaker is, but one of my students, I'm assuming. So you could isolate the sea slug and record whether or not, so the only issue with that process is that if you isolated the sea slug and recorded it, sea slug is still an animal, it is still taking in oxygen, it would be a little bit difficult. And so this is a good question. So Afani wants to put one in sunlight without access to food. And then we have, oh, that's okay. So, and then we have Jessica's asking, does it need light? Maybe we could put one in the dark and don't give it food. So what would be the difference between those two? If it's photosynthesizing, what is the one in the light going to be able to do that the one in the dark won't be able to? Live longer, yes, excellent. Assuming that's Maria. Right, it can live longer. Okay, so we go back here, and scientists found that sea slugs with chloroplasts could survive up to 9 months with no food if exposed to light on a regular basis. 2 months they could survive with no light, which means it was actually getting food from the chloroplasts. Now here's the deal. The chloroplasts, they died off in those 9 months. That's the only reason the slug couldn't survive longer, is because it could only keep the chloroplasts alive for so long. Chloroplasts have a limited lifespan. To create chloroplasts in a plant's body, the nuclear genes inside the plant will build new chloroplasts. Well, that's the thing, is that apparently it doesn't have chloroplast building ability, just maintenance ability. It doesn't have the materials to build new ones, but once it has them, it can keep them alive for their whole lifespan. So that's an interesting piece here. But now we have a new question. How does it photosynthesize? I can't photosynthesize. It shouldn't be able to. So now we have to ask, where does the sea slug get the specialized proteins to maintain the chloroplasts? From the context of this lecture, can anyone answer that question? Where does it get the specialized proteins? Where does it get the genes to make the specialized proteins? From the algae. Well, Eberate, how did the algae give it the genes? That's a good answer though. Got it from the algae. So how did it get in there? There you go. Fanny was listening. Horizontal gene transfer facilitated by bacteria. Okay? So the labeling, this calls fish labeling as a way of genetic analysis. A horizontally transferred algal nuclear gene got into a sea slug chromosome. Well, that's interesting. Algae and sea slugs obviously cannot mate to share their genes. So how did one gene get into the other? It was bacterial plasmid transmission. So there is a YouTube video. I will post this one along with my video here. The key here is that horizontal gene transfer is rare between eukaryotes. It's very common in bacteria. But if a bacteria infects one species, let's say it infects the algae, and it picks up some of those gene bits, randomly picks up the gene bit to maintain chloroplasts. This is a random event. It was not planning to pick those up, okay? Then, when the slug ate the algae, it infected the slug and dropped off those gene bits when it infected it. So that is an interesting, weird way that horizontal gene transfer can happen. Is just picked up by bacteria in one species and dropped off by the bacteria in another. This happens all the time. You probably have little bits of bacteria in your genome, little bits of bacterial DNA floating around, maybe even incorporated into your genome. But nothing happens with them because random bits, we don't necessarily do anything with them. But there has been some studies that have shown that horizontal gene transfer like this happens all the time. Every once in a while, you get a weird thing like this, where suddenly a horizontal transfer leads to speciation and leads to an adaptation to the environment. Yes, the newly acquired gene can be passed on to the sea slug's offspring because it's in the chromosome now. When the chromosome divides, it keeps that. Same with any GMO. The same process happens. You take a bacteria, you induce it to take up a plasmid. There's a couple of other steps there. We basically have created these special transfer RNAs that will move the plasmid that we want to into the plant DNA. But you take plant cells and you infect them with bacteria, and then the bacteria will about 50% of the time drop off that DNA bit. So, and then the plant will incorporate it into their own DNA, and then the plant now has the ability to produce this bacterial toxin to keep bugs away, which it didn't have before. Okay, so interesting piece here. I talked about this in my class before, my Bio 1130 class. So if you are in that or have been in that, you've heard about this slug, but this is the horizontal gene transfer piece, okay? All right. So we're going to pick up more of the bacterial mapping and importance in chapter 17. I just really wanted to kind of introduce that because in the next few weeks we are going to be talking about DNA analysis and recombinance, how we use bacteria as factories to create new proteins. So I wanted to clarify some of the pieces of how that might happen in terms of bacterial DNA. Okay, so that's that one. I'm going to exit of here and go here. All right. Now, we are going to be going through next chapter 9. Okay, I'm going to start with chapter 9. We'll move to chapter 10. We'll probably get through chapter 10 on Thursday, and then we'll move to chromosomal organization. Okay. But chapter 9. The first half of chapter 9 is a lot of the stuff we went through earlier in this semester. Chapter 9 is about basically how do we figure out that DNA is the hereditary factor. So it talks about Avery and MacLeod, and talks about the transformation principles, and it talks about Hershey-Chase, and it talks about all these kind of things. Okay. We went through a lot of that at the beginning, and you should have that knowledge from 1140. Refresh your memory, read through some of those areas, but that's not the main focus here. Instead, I want to be talking more about the chemical nature of DNA and how that makes it a good information molecule. Okay? All right. So all genetic information is encoded in the structure of either DNA or RNA. RNA is a genetic information molecule, okay? And RNA is important in transcription and translation. We'll be going through a bit of transcription and translation in terms of when we're in context of talking about gene expression, because sometimes genes are not expressed. So we'll go through that a little bit later this week and next week, okay? But the idea is that this is the information pathway. DNA replicates inside the nucleus. This is for eukaryotes, but this happens again also through in prokaryotes. Information is transferred from the DNA to an RNA molecule. The RNA transfers that information to a protein, turns RNA into a protein through ribosomes that read it and translate it and put the protein together. Okay? So that specifies the amino acid sequence. We'll go through transcription and translation soon. This is the central dogma. You should know this pattern. DNA to messenger RNA to the ribosome, and then transfer RNA to put the protein together. Okay, so here you have this information. DNA is transcribed by mRNA. rRNA and tRNA take information. So this is rRNA helps build the ribosome. tRNA gets information from DNA to create individual amino acid groups and to attach to amino acids. The mRNA tells the ribosome what it needs. tRNA brings what it needs. That's the translation. Proteins are created. Very brief overview. We'll go through a little bit more of the details in terms of gene expression in particular. But I want to talk a little bit about special information pathways. In some viruses, information is transferred from RNA to DNA or to another RNA molecule. And guess what virus I might be talking about? Anybody? We have a virus you may have heard a lot about. It does not have DNA in it. It is an RNA-based one. Yeah, the one now. HIV is also an RNA one. We're going to talk about both about COVID and HIV right now because it's relevant. And guess what? We'll talk more about it in our recitation class because that's what we're going to be learning about. We're going to be learning about the 1918 flu and comparisons to flu today. Okay. E. coli is not a virus. So it is bacteria. We're going to talk about viruses. Viruses are weird. So we're going to talk a little bit about the weirdness of the viruses. RNA replicase is used in COVID-19. COVID-19 is not a retrovirus. I'm going to introduce retroviruses real quick, okay? But the concept is that RNA serves as the template in reverse transcription using enzyme reverse transcriptase. Okay. So reverse transcriptase is an RNA-dependent DNA polymerase enzyme. Basically, it's using DNA and the structures in another cell to build its own thing. So this is reverse transcriptase. So I'll fix this. It shouldn't say retro, because COVID-19 is actually not a retrovirus. Retroviruses use DNA to replicate themselves. That's why HIV is actually such a difficult virus to cure. Once it's in your cells, it's in your DNA. It actually puts some of the pieces into it. That's what a retrovirus does. COVID-19 is a RNA-containing virus, and it uses RNA reverse transcriptase to basically write new RNA, and to use that RNA, it uses the structures in the cell, the ribosomes and the amino acids and the tRNA in the cell to build itself again. And so this reverse transcriptase is RNA dependent. It's a DNA polymerase enzyme. So in retroviruses, DNA can be incorporated into the host cell genome. So you can actually take the RNA information and incorporate it into the DNA, which is what HIV does. Okay. And when transcribed, the copies of the original retroviral RNA chromosomes are produced. Okay, so when it's read, it makes new retroviral RNA. So that's in the transcription process, creating the mRNA. Well, that's actually the virus RNA. So when it incorporates in the DNA, it's called a retrovirus. So that's HIV. COVID-19 is not a retrovirus. I'm going to show you how COVID actually reproduces in our body. Hopefully none of you have gotten it, but information in case you wanted to know what's happening in your cells. It releases its RNA, so it has a little protein structure, okay? It lands on our cell, it injects the RNA into there. Doesn't touch the DNA. What it does is it uses our cells' ribosomes to be read. It does use reverse transcriptase, but it only uses it to basically create new copies of the RNA using the process, using the system in our cells. Okay, it doesn't incorporate into DNA like HIV. That's why HIV again is so difficult to solve, is because once you have it, it's in your DNA. It's almost impossible to cut it out. Okay, here's what it does. We have RNA. It's a virus. It has a spike protein. And the spike protein uses to basically gain entry. Okay, sort of like it jabs us. Our cells recognize it as something that should come into the body. Now, this is not a great thing, but this is how the virus has recognized itself, okay? It releases its viral genomic RNA, and it uses the ribosome. Now, viruses aren't technically alive because they don't have cells. They cannot replicate themselves. They actually have they only have a few enzymes in there, one of them being that RNA replicase, okay? What it does is it releases that. The host ribosome will read it, translate it, and part of that translated piece makes RNA-dependent RNA polymerase, okay, which then creates RNA. That's the inside of the virus. And some of it is read, and it creates the polyproteins to create the outer shell. And then those are assembled, and the virus replicates and kills off the cell that it used. Okay. So I wanted to explain a little bit how an RNA-based virus might work and exactly what it's doing in our cell. It's not a retrovirus. Retroviruses are indeed only things like HIV, and there's not a lot of those. and the HIV-based viruses are very, so retroviruses are very complex. Bray has asked how viruses are able to mutate. Well, virus mutation is sort of complicated. Well, not that complicated. The deal here is that every time RNA is replicated, there can be an error in there. And this is how it happens in our cells too. Anytime we replicate our DNA, we can have an error. Sometimes the errors don't matter. Sometimes it doesn't affect us at all. Sometimes those errors build up and can lead to an offspring that the something bad or maybe something good, maybe an adaptation. A random DNA error or a random RNA error. So because there's so many of these virus particles and there's so many bits of RNA, any errors that happen, well, it's in a small percentage of the population. And that small percentage of the population, if it's something good, perhaps that mutation makes them resistant to an antibiotic. It changes the shape of their spike, which is what we are targeting right now during the creation of a vaccine. If that spike changes shape due to a mutation, well, we can't kill off that portion of the COVID virus. But again, unless there's a selection pressure until we actually have something that kills it off, then that's not going to impact it. It is why you have to get a flu. So the flu itself is actually DNA based, but it's the same process. It's why we have to get a flu shot every year, because the variants that are created, they survive and can continue to infect people who have gotten the vaccine. That's why then they became the more prevalent one, and that's why we have to get a flu shot next year. So we just slightly modify the vaccine each year. Since this is a novel virus, we don't have any vaccines that work on it. And we can, you see how this spike protein, it is recognized right here. Well, we can actually tag these people who have gotten COVID before, can tag them with proteins, which tells it to be eaten by a white blood cell. Okay. This is actually a great video. Let me pull it up. Hold on. XKCD. There we go. So this is a good one. Let me see if I can. Okay, we'll go. So I'm worried about humans developing resistance to us using pasta. Human immune system is a nightmare. It's the worst. It's the scariest thing in the universe. Who wants a hug? We can only survive by staying ahead of it, jumping from person to person, keep mutating and evolving, but now humans are adapting too fast. They spread through the water, they built pipes. We use mosquitoes, they put out nets and poison everywhere. They spread through sex, suddenly they had all these plastic things. This time we thought we had them. One of us got real good at transmission through everyday contact. It was great. We were tearing through lungs, spreading like wildfire. I hate lungs. All of a sudden, humans everywhere just stopped. Stopped working, they stopped seeing friends. They're just sitting there in their houses, washing their hands. Alright. Suddenly, humans have become dead ends. We try to jump from one to the next, but there's no one to jump to. We can't escape. We're trapped in there with those ghastly immune systems. Again, our immune systems, once they... that's why we get that terrible cough, is because our immune systems are fighting terribly. That fever is partly due to kill the virus. This is why people with suppressed immune systems actually have such trouble, because what will happen is that the virus, it infects the lungs, and with no immune response, pneumonia can develop very fast before you feel sick. So this is why people with good immune systems actually they'll get sick fast and then get better fast. Okay. All right, so we're trapped in there with those ghastly immune systems. Even if we win in a fight, there's nowhere to go. By staying inside, humans have become resistant. How could they evolve that fast? Humans take decades to reproduce. Okay, so the idea is that humans start looking at their phones, talking, writing words, and making signs. A human named Gloria Gaynor filmed herself singing at a bathroom sink, and they bought lots of pasta. Around the world, they all went home, started washing their hands. They saw what they were doing and changed their behavior to stop them. Brains are the worst. They can't sustain this. They must be bored and tired. Will they give up? I don't know. They seem determined to protect each other. And they have a lot of pasta. This is one of the things where, again, slowing the spread and such, but this is the idea is that that's they move from person to person, and certain ones die off and certain ones survive. The surviving ones have a mutation that may be beneficial, and that continues on. Okay, keeping them isolated prevents this from happening. The jumping from person to person encourages the mutation. Mavis has asked, why is it that people who have had the infection do not become immune to it? No, this is still under study. This is a new thing. There's been some scary articles going around where someone who got the virus did not tested negative and then tested positive later. They weren't having symptoms, though. We don't actually know if this is a fluke or not. And the hope is that we can become fully immune to it. The question would be whether or not there's a variety of viruses in there. Some of them, so you test negative because you have such a small viral load, and then you test positive because you have a very, later because the virus load increases. And I don't know. That's up to, this may not happen all the time. There's only been a couple of isolated cases of that happening. But good question, and something that to be slightly concerned about, but understand that it's still in the early stages. Okay. So RNA-based viruses. All right. So we're going to talk a little bit about the characteristics of genetic material. A lot of this should be review, but we're trying to kind of link it to genetics overall. Okay. So four major characteristics are needed for a molecule to serve as genetic material. It needs to be able to replicate. It needs to store information. It needs to express information. And it needs to allow for variation through mutation. And that's, again, how all viruses and bacteria and humans evolve over time. And again, that variation by mutation, that happens, we talked about the various types of mutation earlier. It needs to be able to express that information. Viruses are a weird one because they don't have the ability to express it except in other cells. But we need to, genetic material needs to replicate, be stored, and express. Okay? We're going to talk a little bit about the base knowledge necessary for the next few chapters. You hopefully learned most of this in Bio 1140. If you didn't, please review the first half of the chapter. Okay? Basically, they talked about Griffith's observation is virulent versus non-virulent cells. What are they transferring? We learned this is the bacterial plasmids of these virulent cells, and transformation is happening. It could only happen, though, if virulent bacterial cells had intact DNA. You destroy the DNA, and there's no passing of virulence on. Okay, so this is a key to pointing out what the inherited thing is. Hershey Chase showed that bacteriophages are injecting something into bacteria to direct their components. They marked components and only DNA passed on, not the protein. The protein came from the host cell. So this gives us more information that it is DNA that is the genetic material. Again, this is covered in other classes in detail. But this is the basics of the Hershey Chase. Okay, so we had the protein coat, phage DNA, and these fibers. They found this genetic material was injected. They tagged all the parts of the DNA, and they were able to find that only the DNA was consistent from here to here, but the protein was new. The protein stayed on the outside, did not pass to the inside. All right, so the structure of DNA does hold the key to understanding its function. We can understand better how how DNA is expressed through understanding how it is structured, okay? The structure of DNA was such a breakthrough that we talked about near the beginning of class because understanding genetics, genomics, epigenetics, gene regulation, biotech, recombinant DNA we have to understand the structure of DNA in order to do any of that. And that's why it was such a short period of time between, so we have 1950, we discovered the structure of DNA, and by the 1970s, 1980s, we're cloning sheep. And that's how fast it progresses after we actually understand the structure. Structure is extremely important to understanding molecules. I know some of you guys are taking biochemistry right now. And so part of that is understanding the structure of DNA. All right, so there's 3 levels of DNA structure. There's the primary structure, there's just the sequencing. Okay, so the sequence is not quite enough to understanding how DNA works, but we can get the nucleotide sequence. Then there's the secondary structure, this helix that keeps them together, that double helix. That's the secondary structure. And then the tertiary structure, we will be talking about this either later this week or early next week. The higher order folding of DNA. Okay. So we're going to start with the DNA nucleotide and work our way up. And again, we'll get through what we can today and continue this and DNA replication on Thursday, plus maybe talking a little bit about chromosomes. Okay. So deoxyribonucleotides, each nucleotide consists of 3 parts. So you remember what each nucleotide contains? What does each nucleotide contain? All right, nitrogen, phosphate, and sugar. Okay, so deoxyribonucleotides contain deoxyribose, not ribose, but close. Good. Okay. So we have a sugar. Again, deoxyribose for DNA and ribose for RNA. That's the difference between them majorly. Okay. A phosphate and a nitrogen-containing base. Okay, so that's what we talk about when we talk about bases, base pairs. We're talking about specifically that's where the information is contained, because that's the part that varies in the line here. Okay, good. You guys all know this. Who is IRS of the government? Must be someone else who has a Twitch account before this. All right. Okay. That almost makes sense, Rana. Okay. The sugar is a 5-carbon sugar, pentose. Okay. As I said, the ribose versus deoxyribose. This extra OH group is important for ribose, right? RNA is very complex and very reactive, in large part because it has that extra oxygen on it. The lack of oxygen actually on deoxyribose makes it a more stable molecule, good for storing information, not so great for doing anything else. Okay, so phosphate groups can carry a negative charge that makes DNA acidic, also makes DNA negative. Okay, so this negative charge, that's why we call it deoxyribonucleic acid. Again, if you are in biochemistry, you are learning many of these things that we are talking about. This is a review and not, this isn't genetics. This is biochemistry, but it does relate to genetics, okay? So we have a nitrogen-containing base. We either have a pyrimidine or a purine, okay? So pyrimidine. The pyrimidine is a single carbon and nitrogen group here, because this is the nitrogen-containing base. We either have cytosine, uracil, or thymine. Uracil and thymine, very similar, but you will note a little bit of difference here, right here. Okay, H3C instead of H. Okay, that's the one that interacts with RNA, and is this is the one that interacts with DNA, and this is the one that interacts with RNA. Okay, purines. There's 2 purines. Guanine and adenine. Okay, guanine and adenine are larger, again, C, G, A in both DNA and RNA. Thymine in DNA, uracil in RNA, and that has to do with their different chemical structures. Basically, RNA can't interact with thymine. All right, one thing I want you to remember is understanding a little bit about, remember your purines and pyrimidines. This is, I think it's a little bit much. I remember it, so you can remember that purines are pure as gold. Purines are adenine and guanine. Pure as 2 gold rings, remembering that they have 2 rings, not just 1. Pyrimidines, so we have UC tombs. Okay, so it's like a pyramid, pyrimidine. Okay, or cut the pie, whatever helps you remember. I remember them this way. I remember that purines are big, and pyrimidines, which is a long word, is small. You just remember that they're the opposite size, and then you can remember that CUT, cut the pie. That helps you remember that cytosine, uracil, and thymine that are the pyrimidines, and the purines are adenine and guanine. Whatever helps you remember it. Okay, you do need to know this because it does have to do with how they are connected inside of the double helix. I'm going to show you. All right, so you have adenine and guanine. Okay, those are the pyrimidines. Those are the purines. Thymine and cytosine in DNA, and those are the pyrimidines. All right, so this is just showing here overall how they look on there. What you can see here is that the T and the U, again, they attach a little bit differently, in large part just due to this interacting OH group this interacts a little bit differently. Okay. All right. So we have the phosphate group that acts as the backbone. We have a sugar phosphate backbone. The nitrogenous bases stick off of the edge. And they each have these hydrogen and oxygen sticking off them. That is how they interact with other bases, because we form hydrogen bonds, which are easy to break apart. Think of them as like Velcro or a zipper, easy to pull apart. The DNA double helix is set up so that, in this way, the double helix and that shape makes it so that it's evenly spaced all the way down. Okay. So these are 2 anti-parallel, complementary DNA strands. Now, I mentioned this because we will be talking about DNA replication and DNA. So you have to know. So you should have learned in the intense detail how DNA replicates. We will go over it briefly. I do not want to be testing you heavily on all of the different enzymes that encourage replication. You should know this. If you do not know it, well, I will share a couple of videos that will help you know it. Okay. So, 2 anti-parallel complementary DNA strands. Basically, you can only add new bases to the 3' end. That has an OH group. It is reactive and can be formed through dehydration synthesis. You have OH on one, H on the other, bonded together, H2O pops out, and you've put them together. Okay. So the 5' end of one strand is opposite the 3' end of the other strand. And so they're linked by these strong covalent phosphodiester bonds. The phosphodiester bonds are very strong and are not easily broken apart. Okay, but what is easily broken apart is the inside. You're able to open up the interior easier than breaking the actual strand. So these stacked in here in nitrogen bases. Hydrogen bonding between complementary base pairs on opposite strands. Okay, this is part of the important bit. Each new nucleotide, again, can only be added to the OH connected to the 3 carbon. The 5' end is unreactive. The 3' end is reactive. You can only build DNA on the 3' end. This matters for when we're building DNA in particular because directionality matters, okay? Because it's double-stranded. You can only grow from its 5' end away towards its 3' end. The 3' end, it can only extend it. This matters a lot, and we're going to talk about it as we talk about DNA replication. One thing you're going to notice is that T and A pairs have 2 hydrogen bonds. U and A also have 2 hydrogen bonds. RNA is very rarely double-stranded. And then G and C have 3 hydrogen bonds. So this is the only way that you can have an equal size pairing between these two, is that it has to organize in that shape, and that also helps with the twisting strands in that way. Okay. C and G pairs have 3 hydrogen bonds. And here is the famous photo 51 I've showed you already that showed the estimate of the periodicity of the nitrogenous bases. And because of that periodicity, they were able to estimate how the bonding happened. So different base compositions come from different sources and ratios of bases. Some have, so their A to T ratio is different from their G to C ratio. Remember A and T. Okay, so these ratios vary, okay? But generally, A will equal T, G will equal C. In DNA, the amount of purines, A plus G, equals the amount of pyrimidines, T plus C. And yes, T and A break either than CG. TA breaks either than CG. Absolutely. We're about to go through that. Okay. So we talked a little bit about Chargaff's rules before, is that Chargaff used chromatographic methods to separate the 4 DNA bases, and he basically measured how much A, how much T, how much G, how much C. Okay? Amount of adenine is always equal to the amount of thymine. That makes sense. Amount of guanine is equal to the amount of cytosine. That makes sense. The sum of the purines is equal to the sum of the pyrimidines, because a purine must always be bonded to a pyrimidine. Also makes sense. The percentage of G plus C, okay, so so we're talking about, so G plus C, G and C are equal, but there may be more G and C in one DNA than A and T. Those aren't equal, okay? So the 2 pairs are not equal to each other necessarily. You could have more G's and therefore more C's, more A's and therefore more T's, but so those don't have to be equal. But I want you guys to figure this out. If you had the percentage of T, can you figure out the percentage of G? All right. So A plus G equals C plus T. A plus T equals, I wrote that wrong, didn't I? Okay, A equals T, C equals G. And it should actually be A plus T equals C plus G. I wrote that wrong. I will fix it. All right, so you find out that a DNA molecule is 23% thymine. Okay. So if you have 23% thymine, how much adenine do you have? $23. Great. Good. So we now have accounted for a total of 46% of the rest. Okay? So 23%. So you've now established that 46% of the DNA molecule is A and T. The other 54% is C and G. Well, now we know C equals G. So if C equals G and you have 54%, how much is G? There you go, 27%. So guanine, if you have the percentage of thymine, you have the percentage of guanine and cytosine and adenine. You can tell all of it, okay? So that's what I wanted to encourage. This helps us, the pattern of DNA molecules can be figured out through just one base. All right. So we just talked about this a little bit. Hydrogen bonding between complementary base pairs is relatively easy to disrupt, but the strength of the bonding can vary depending on the nucleotide sequence. Which 10 nucleotide sequence would be the hardest to separate from its complement? I just answered this question earlier. Okay, so I have a couple of B's. And correct. So Asa has it. B has the most G and C. Those bonds, because on the opposite strand is going to be a C, a C, a G, a G, a C, a G, a C. Okay? Those bonds have 3. Slightly harder to break that one than the others. Okay? Again, they're relatively easy to disrupt, but there is more hydrogen bonds to disrupt in this string than in this string. Okay. So what's true of the secondary structure of DNA? Read through these here. Maybe this wants another explanation. I can explain it again, that's fine. So for this one, it's going to be E. Yes. The paired nucleotide strands are antiparallel. I just explained the C plus G does not have to equal T plus A, and that the phosphodiester bonds are actually on the backbone, not on the bases. The sugar phosphate groups are on the outside. That's the backbone, and the nitrogen bases are on the inside. Okay, all right. You wanted a explanation, Mavis, of this one. Why is this stronger? Okay, so the reason this is stronger is because G. So this is one strand of the nucleotide sequence. We're not looking at the other strand, but we can tell the other strand from this. This is C, C, G, G, T, C, A, G, A, C. Okay? This has multiple G and C bonds. So this is G to C, G to C, C to G, C to G. Those have 3 hydrogen bonds each. Well, the A and T, which so on this one above, we have A to T. That is only 2, 2, 2, 2, 2, versus 3, 3, 3, 3, 3, and 2. Okay, so if you add up all the hydrogen bonds, you're going to find more hydrogen bonds in this strand because it's more G to C's. Okay, we're only looking at one strand here, so we have to infer the other strand and know how they are connected. Okay. Again, hardest to separate from is complement. We don't see the complement. We can tell what it is when we see this many G and C pairs. That makes it more difficult. Okay, Mavis got it. All right. So, if 20% of bases in DNA are cytosine, what percentage are thymine? We just went through this, so hopefully you guys can remember how to do this. It is C, because 60% then are A or T. You break that in half, 30% are T. Correct. So if 20% are cytosine, then 20% is guanine, not thymine. The phosphorus carbon backbone is held by phosphodiester bonds. The nitrogenous bases are held together by hydrogen bonds. So it's not 20. It is instead 30. The reason it's 30 is because this 20 cytosine is C, and then G is its complement. So 20% is G. Then you have 60% more. You break that in half, 30% T, 30% A. Okay. So let's talk through RNA briefly, and then we'll stop for the day for our this part, and then I'll continue and go through replication and a little bit about chromosomal structure on Thursday. Okay. All right. Good. Everyone got it. RNA differs from DNA in 3 important ways. RNA is a single-stranded molecule. The pyrimidine nucleotide uracil replaces thymine in RNA. Generally, single-stranded goes through double-stranded stages, but it's generally single-stranded. The sugar ring is ribose. Ribose makes RNA less stable than DNA because of the extra OH group. The additional hydroxyl increases susceptibility to hydrolysis, but it can also react more strongly and do more stuff. Okay? It is not just a storage molecule. So RNA has a nucleotide linked with phosphate chains. The sugar ribose replaces deoxyribose. Okay, we already explained that. Most are single-stranded. Animal viruses have double-stranded helixes. So an animal virus like COVID would actually be double-stranded. But that's just sort of a weird thing with animal viruses. RNAs can be single or double-stranded. It depends on what they are. Okay, so we have rRNA. That's the ribosomal RNA. It is the structural complements of ribosomes for protein synthesis. So rRNA is inside of ribosomes. And all things that create proteins need ribosomes. So RNA is very vital for that. Okay. mRNAs are the messenger RNAs. They are the template on which genes are written. Genes from DNA are written onto them, and they take so mRNAs go into the nucleus, grab the information, take it out of the nucleus. They carry genetic information from the gene to the ribosome. Okay, they are the architects taking the information to the builders, which are the ribosomes. The tRNAs are the transfer RNAs. They carry amino acids. They're basically this special structure, and they are able to snag amino acids that are free-floating in the cytoplasm, and they carry them to the ribosome for protein synthesis. So reactivity links them all. Ribosomes have to be reactive because they interact with mRNA, tRNA, and they and they put together proteins. They have to have reactivity abilities, ability to use energy. mRNAs, same thing. They have to be able to react and change and move, and same with tRNA. The key here is that DNA doesn't do any of these things. It is just a template. It is just sitting there. It is a stable storage, but it is not able to do much without the help of reactive RNA. Can't even replicate itself. Special secondary structures can form in DNA and RNA in particular. This is a picture of a tRNA. It has this loop structure, a small double-stranded section. So you see this Bio 2030 recitation. We're gonna pause here and then come back at 3:30. But these hairpins. Hairpin structures on DNA can form, and also on RNA. RNA is forming this structure because it is interacting with a specific, down here it has a specific chain that it interacts with on the ribosome, and it grabs a specific RNA, a specific amino acid up here. All right, and RNA can contain numerous hairpins and fold up into complex structures, and this can affect transcription. We'll talk a little bit more about this when we talk about gene expression as well. Okay, we're going to pause there, and I'll stop the stream, I'm going to put the link. Go back to there and click the link in Blackboard. I will pull it up on screen here. Let's go back to GCC and Blackboard. So you're going to go here to Bio 2030. And you're going to join the Zoom meeting. Click on this here, okay? And that's going to open Zoom. If you don't have Zoom downloaded, it will prompt you to download Zoom. So I'll give you a couple minutes to set that up. I have to reorganize my office here, but I will see you guys there. And again, this is not required. I will record the Zoom session, and I will post the PowerPoint that we went through, plus the set of questions necessary. You will be turning something in by next week, and that thing that you turn in will be based mainly on this Zoom session. Again, if you don't have the ability to participate in the Zoom activity, I'll be able to hear you guys at that point, and we'll write stuff up on the whiteboard that I have in my office. If you can't participate, then that's okay. But you'll have a little bit of catching up to do, and I will try to post that as well on online. I will be recording it, and you'll be able to follow that on YouTube as well. I'll post this YouTube video soon. Okay, that's all I have for now. I will see you guys in a few minutes. Again, come to the Zoom meeting if you want to talk to me more, okay? I'll turn on the Zoom meeting pretty quick.